Well, welcome everybody, and thank you so much for taking the time and trouble to come to tonight's meeting. This is the first meeting of the new presidential year of the Nottingham Medkai, and it's a pleasure for me to be the president and be elected to the presidency for this year. First of all, just a little bit of housekeeping business. I should say right at the beginning, the PGMEC has been refurbished. And we've still got one or two, it's a beautiful refurbishment, we've got one or two glitches, mainly the lights, which may go up and down randomly during the evening. We did manage to get them down to an appropriate level early on, but they've just suddenly now gone up on their own. So bear with us, we'll try and adjust the lights. Some business matters. Could you please sign the sign-in sheets outside the room? I'm sure you've already done that, but if you've forgotten, if you can sign on the way out too. The charity for this year is Life Cycle 6, which is the University of Nottingham's charity which is supporting breast cancer research in Nottingham. Clearly that's very close to my heart, and some of my excellent colleagues will be involved in receiving some of that support. So it is very close to what I'm doing, but please be generous and support Life Cycle 6, and there are some leaflets outside there, and I'm happy to talk about that at length if you wish to later on. Apologies have been received from Dean Temple, Claire James, Adrian Manhire, Nigel Sparrow and Charlie Charlton. I do have the sad duty of announcing the deaths of three former society members, Paul Jacks and Neville Smith, and also our former president, Rami Seth, who was president between 2002 and 2003. As I suspect many of you were at his funeral, it was very well attended, and it was a fitting celebration of his remarkable life. He was a true polymath and fantastic character who I'm sure we all loved and enjoyed meeting with him informally and formally in his presentations to the society. If we could just take just a few minutes of silence, or a few moments of silence. Thank you very much. The last meeting was held on the 18th of May 2016, and the minutes are outside if you wish to read those. There's also, well, I was going to say there are copies of the programme outside, but they've all been taken already. Catherine's trying to find some more, but our room has been locked and the key seems to have disappeared. But there will be programmes available, and Catherine can send them if you want to ask her later on. I'm sure she can provide those to you. There are also electronic versions available too. There's a couple of things happening fairly soon. Helen Percival is organising a charity cycle ride on the 16th of October to support Life Cycle 6. If you're interested in cycling and keen to get involved, then please see Helen as soon as possible, and Catherine has sent a notice around about that. But that's just an open event for anybody who's interested in supporting that. The next book club meeting is on the 17th of October, and Chung's charity dinner is on the 23rd of October. It's always a sellout, so please try and get your booking in as soon as possible. It's now my pleasure to bore you stupid with my life and my thoughts on the presidency and the Nottingham Medkai. As I'm sure you're aware, I'm a Nottingham medical student, and I've lived in Nottingham now since 1973, although I still see myself as a Mancunian. I think it's your sort of former years where you grew up sets your scene for life, but I've lived in Nottingham longer than any other city, and all my children have been born and brought up in this city. I am a Lancashire, Cheshire lad, although interestingly my name Ellis, the National Trust used to have a website which you could actually look at how your name had progressed over the years through the country. And Ellis is an Elijah. It comes from France originally, crossed the Channel, and then migrated, the majority to Wales, interestingly. And I was assumed I was a Welshman historically, but I've discovered, and this is difficult for a Lancastrian, but I've discovered that there was a small tribe of Ellises went to South Yorkshire. Into the Denbigh, Penistone area. And so in doing my ancestry, I've discovered that I'm actually a Yorkshireman. And you can imagine that somebody growing up in Lancashire, and as somebody used to say, that your nearest neighbour is your enemy, everybody else is just a foreigner. I remember there's one of the comedians in Scotland saying, everybody from Edinburgh is just the enemy, the rest are just foreigners. And it's a bit like Lancashire and Yorkshire. But I was brought up in Lancashire. My dad was from Chorlton, this is his family. My father's the young chap just down here. He met my mother in Scotland, in Falkirk, so I've got a very strong Scottish connection in my life. They met ice skating, and I'll come back to that later on. But this is my mum and this is my dad in an ice pantomime when they first met. I came along quite a little bit of time after that, after they'd migrated around the country with various jobs. And I was born in Edinburgh. At the age of 6 months, we migrated to India on one of the ships that took people out. Aeroplanes weren't really very common at that time. My parents obviously had a party during that. This is one of the, I think this is New Year's Eve, and it's a fancy dress Mad Hatter's tea party. And that's my dad there and my mum dressed up on the cruise ship. I have no idea where I was at that time. My father, the reason for going to India was my father worked for ICI, the chemical company, and he was setting up a dyestuffs factory in conjunction with an Indian company in a village called Balsar, 200 miles north of Bombay. That is a thriving community. It's actually the most famous town or village in the world for mangoes. It's where the Alfonso mango comes from. My father still recalls the smell of mangoes for several weeks as they were all shipped out of the town. And we used to get mangoes sent to us when we came back from India, and I still adore them, I have to say. This is my mother opening the factory right at the start of our life in India. We returned back when I was 4.5. BOAC had been established, and we went back by plane rather than boat. And I was a junior jet club member. Unfortunately, I've lost my membership card, and BOAC no longer exist. And we moved to a village called Bramhall, south of Manchester. And this is the family home. No, of course it's not the family home, but this is Bramhall Hall, which is a Tudor house there. It's a beautiful park. If you're ever in that area, do call in. The reason I'm showing it is that my mother, there's a beautiful park associated with the lakes, and my mother used to take me ice skating on those lakes every year. She'd check diligently if the ice was thick enough to skate, and we'd go out and skate. I don't think those lakes freeze up anymore, and that's an interesting observation of life, that the climate when you're younger and things you can remember is very different, and that's why one of the talks we're having is around climate change and how we can prevent it, because I've certainly experienced those changes in my lifetime. This is me as a young lad back in the UK. I was fortunate and privileged to get a grammar school education at Stockport Grammar. This is a school as I remember it. It's very, very different nowadays. It's a co-educational grammar school and semi-public school. But I had some very happy years there from the junior school all the way through my career. What did I get up to as a child? Well, there's a curious sport in the northwest of England called lacrosse. It's migrated over from the sort of the Ivy League universities in America and in Canada. It was a North American Indian sport. But the schools and young men in Manchester in the northwest played lacrosse. And for some reason, our headmaster, although we played football in the junior school, in the senior school you could play rugby or lacrosse. Now, I'm an August baby, and so I was the smallest and youngest person in the year. And the last thing I wanted to do was go and play rugby with the largest and oldest blokes in the year. So I chose lacrosse, and I played lacrosse as a sport until I was 45, when my knees were beginning to pack up and swell at games. Tony Marsh also, who's a GP in Nottingham, and I played many, many games together. He actually lasted longer than me on the pitch, but I continued to play for Nottingham University until I was 45. It was a great sport. The problem is that the ball moves so fast that it's never been a television sport, so it remains a minority sport. But that's my sporting pastime and great fun. I mentioned my Scottish background. My father's cousin he was an architect in Edinburgh, and he designed one of the skiing lodges in Glenshee. It was the time the Forestry Commission were buying up a lot of land in Scotland, and they were selling off the houses but keeping the land for forestry. And he acquired a castle for a few quid, because they essentially wanted to get rid of it. And it was a nightmare sort of thing to have. But he said to my father, "You can use the castle anytime you want, free." So, of course, my father, Yorkshire background, Lancastrian and Scottish background. Every holiday from the age of 7 to 30 was spent there. All my friends from medical school, all my school friends have been there. We stayed in either parts of the castle or down. And so I have a very strong affection for parts of northern Scotland. And I learnt how to fish there. And one of my passions is fly fishing for both trout and salmon. And this is me fishing recently, or every year I go up to the very north of Scotland to Sutherland to fish on a river called the Castley. Chris Elston never believes that I catch any fish there. Sadly, he's away on holiday today, but this was just some proof of a fish I caught this year in the river. Now, this is a hidden past, and I think none of my friends know a great deal about this, but this is a budgerigar. And I used to like pets, and I used to keep pets, and eventually I migrated to budgerigars. I don't know why, but I did. But I took to it with a passion. I built my own aviary when I was about 14 or 15, and I started breeding budgerigars. And I did actually do quite well. This is a cutting from the Stockport Advertiser, and there is Mr. I.O. Ellis, who is the junior champion cage and aviary birds. I only managed that once. But the reason for talking about this, although for me it was great fun, is that the colour patterns in budgerigars are immense. It's incredible. This is just a snapshot just showing blues and yellows, but there are greens, greys, and all sorts of variations of those. And I got very sort of, for one particular reason, I wanted to breed a budgerigar which was a cinnamon powder blue. Now that sounds a bit weird, but that's what I wanted to do. And as a kid, you often think that, and I thought, how can I do this? So this is something like a cinnamon powder blue. What you have to know about budgerigars is the colours are obviously genetic. They're due to gene mutations. Some are recessive, some are dominant. The two dominant genes are grey and green. Getting powder blue cinnamon is about the most recessive thing you could possibly do. So it's virtually impossible to do, and I never managed to achieve it, I have to say. But I did read a book on the genetics, and this is, I got this from the library, this book, but and I wish I'd bought it because it's a fantastic book. It just goes through all the genetics and variations and how you would achieve the colour of a bird that you wanted to get. And it's a very complex process. It's interesting. If I'd bought this, I just looked it up on eBay recently. It's currently going at old versions at £2,000 because it's the only book of its type in the world. The reason for mentioning that is that I began to understand about genetics through budgerigar breeding. It sounds bizarre. But when I decided to go to medical school, I travelled to a number of medical schools, but I ended up choosing Nottingham. And two reasons for that. One is that they offered me a place, and the other is they were so welcoming when I arrived. The entrance exam, or the entrance process for Nottingham, there was an interview, but there was also, you were met by two medical students who took you around and showed you the university. It's a beautiful campus university, and I was sold, much to my mother's dismay, because she wanted me to go to St Andrews. But at my interview in Nottingham, one of the interviewers was the professor of genetics. And he looked at my CV and said, budgerigar breeding. Now that's interesting genetically, isn't it? So we had a long conversation about the genetics of budgerigar breeding. I think that's probably the reason I passed the interview, because I think I was pretty hopeless at everything else. Anyway, this is our intake. I was a medical student in 1973. There are 48 of us, so still a small year. Jim is and Henry are both here, both members of the year. There's little me, looking very young and innocent at that time. I think I was the youngest in the year because of my August birthday. Medical school years were reasonably uneventful. There were some successes and some failures. I managed to fail one or two exams, but I did manage to creep through. One or two anecdotes. I don't know if Miss Baker's here, but Jim and we were both medical students with Miss Baker. We were actually medical students on a number of rotations together. With Miss Baker, there was two curious aspects of that attachment. One is that there was a very hierarchical system on the ward round, and the medical students were at the back of the ward round procession as we went round. But there was also the senior registrar who was also at the back of the ward round procession, and this is a guy called Bruce Gilbert, or Bruce. He was an Australian, and at some point, we never discovered why, there had clearly been a difference of opinion between Miss Baker and Bruce, and he'd been relegated to the medical student end of the procession. The other thing we learned was that when you went into an outpatient clinic and opened the door and met a new patient there, we learned very quickly there are two questions you had to ask. The first question was, do you sail? And the second question was, are you a doctor or are you medical? Or is your husband a doctor or are you medical? Now, if the answer to either of those questions was yes, you immediately said, thank you very much, Miss Baker will be in to see you in a minute, and left the room. Because we quickly realised that Miss Baker had several patients, or groups of patients, particularly all the medical communities, wives and themselves went to see Miss Baker, and also her sailing club friends all came to see her too. And we made sure that we're not involved in those, with those patients. Lots of other anecdotes, and I'm sure Jim has many, many as well. But great fun and a great place to become a medical student. I also met this character. Chris was my tutor when I did my honours year in pathology, and we did our post-mortem examination together and a surgical case together. I want you to remember that this is an old photograph. This is the microscope that we learned to grade on, and hairstyles in those days were a little bit different than they are now. Chris isn't here, so I'll come back to this anecdote later on. This is me graduating. My only achievement as a medical student was to win the North Nottingham Consultants Prize, and some of the people who used to set that quiz, I think, are in the audience tonight. This was based in this building, and every week a radiologist, a pathologist, and a physician would set 3 questions, and you had to answer those questions. And this was over a period of several months. And the only reason I won is I have a dogged determination, and I just managed to keep answering the questions. All my other colleagues, like Jim and others, got fed up of doing it and just left. So this just shows a tenacity rather than any innate skill or intelligence, which I haven't really got. We did graduate, and this is something from our graduation book, and I'll just read it to you. The medical student is likely to be one son of the family too weak to labour on the farm, too indolent to do any exercise, too stupid for the bar, and too immoral for the pulpit. And we felt this was appropriate for our year at the time of graduation. My parents, despite that, were reasonably proud of me when I graduated. So after graduation, house jobs were here at the city with Langman, Banks and Duff, and then the General Hospital with Jack Hardcastle and Patrick Bates and Jim Burke. I was fortunate in that at the General Hospital, it was the time the Queen's Medical Centre was opening, and actually migrated the first adult ward of patients. The kids had already moved from the General Hospital to the QMC. I have to say there's some wonderful anecdotes there. You may or may not remember this, but when Queen's first opened, they had this wonderful way of transporting the patients' food around from the kitchen areas to the wards, and the patients could order their food in advance, and their individual tray would arrive still warm and heated. And of course, in the first week, that completely broke down. And one of the administrators was on the local TV channel saying, well, everything worked beautifully before the patients arrived. And what is the raison d'etre of a hospital? Well, I was originally going to be a surgeon, and that's why I did Jack Hardcastle's job, and it's nothing to do with Jack Hardcastle that I didn't choose surgery. But at the time, there was a huge bottleneck in surgery and progression. John Doran, for example, was 35 and was still a senior registrar. I was just in my very early 20s, and I could see that progression in surgery was not good. There were several people that I knew at the time leaving surgery, and so I went to see Ian Dawson, who was the professor of pathology, and said, what about a career in pathology? And he said, have a job. So no interview, I was just given a job because at the time recruitment in pathology was difficult, but it was great fun. And I started as a lecturer in histopathology in 1980. Great place to learn. The pathologists in Nottingham were fantastic. I have to say Nottingham became known throughout the country as the best place to train in histopathology and cellular pathology, really because of the calibre of the individuals. And that's the reason people like Jim and myself, people say, why did you stay in Nottingham? Well, it's because the progression of our careers and also the teaching was fantastic here. We both said as housemen that we would do our house jobs, start in pathology and then go to London as soon as possible, but neither of us did that. And I think they're a reflection of the calibre of the individuals in pathology in Nottingham at the time. I also had a role in teaching, and I remember one of the things Ian Dawson said to me a few months after arriving is, Ian, it's your time to do a bit more teaching. You've got the alcohol lecture to deliver. Now that's a bizarre thing to do because the alcohol lecture on pathology was given to the psychiatric students in Mapley Hospital just for one session, an hour during their rotation dealing with psychiatry. So every month or two I would trek around the ring road in my old beaten up car with some pots of liver in the back, take those out and deliver a lecture on the evils of drink to the psychiatric students, and also telling them about some bizarre aspects of alcohol, such as Chianti drinkers' disease and other such interesting Quebec beer drinkers' disease. I love to research those odd things. But I also had a raison d'etre in research, which was difficult in histopathology at the time because there was very little research going on. Ian Dawson's group had a small research lab, but it was very inward-looking rather than outward-looking, and they were not really interested in new people coming in. And this taught one of my sort of earliest lessons in life was in trying to start doing some research. At the time, lymphomas were poorly managed or poorly diagnosed in the UK in general because some of the techniques that were emerging weren't being introduced, such as immunocytochemistry. And Richard Powell, the new immunologist, and Juliet Fowraker, another SHO working with me, we decided in a corridor conversation to try and set that up in Nottingham. This technology. So he said, he'd just come from Birmingham, so he said, contact the professor of pathology in Birmingham and we could go down and learn immunocytochemistry there. So he contacted Professor Curran and got a letter back from Professor Curran saying, under no circumstances can these two individuals, this is verbatim in this letter, under no circumstances can these two individuals set foot in my department. If they do, I will not examine them in the MRC path examination. Now you can imagine as an SHO, being told that by the professor of pathology from another university that they wouldn't examine, you thought your career was ended. Now that's the most awful statement I've ever read, and it sort of led me to believe that there must be some evil people out there, because I just felt everybody was lovely at those days, but they clearly weren't. The only thing I could say by Curran, he did become president of the college, and he was the only president of the college that wasn't knighted, which I was absolutely delighted about. So Richard Powell just said, no problem, go upstairs, because the professor of immunology upstairs, McCallum, will teach you what to do. So we went upstairs, learnt immunocytochemistry, and then came back again. But it taught me a lesson that some people are good and some people are not so good. I have to say that we then went off and worked with David Mason in Oxford, who was fantastic. Just opened the doors for us. We spent 2 days in his lab. He provided us with reagents and constant reassurance. And there were people in the country who were really supportive of young individuals who wanted to progress and do things that were novel and new. There are also things in terms of the diagnostic service and teaching and my education. There was a meeting every few months of consultant pathologists in Nottingham called the Black Box. And the Black Box is where you put in cases where the diagnosis was difficult. And the consultants would meet, the consultants from Derby, Kings Mill, Nottingham, would all gather together. There would be some sherry or wine beforehand, which would make life a little bit more interesting. Trainees were allowed to attend, but could be seen but not heard, unless they were actually asked to make a comment. But these meetings were fantastic, and you really learnt a huge amount of pathology. But there's one case, that Jim may remember this, which both Jim and me actually had the tenacity to actually speak up. Because there's a case of what's called microglandular adenosis of the breast, which is a curious process which is considered benign. And I remember asking Chris Elston, why is this benign? Because there are certain features of it which you associate with invasive cancers, pathologically in a cellular pathology way. But he said, because it is. Now, it's not a good answer. And it's one that's nagged in my brain for years and years and years. Just remember microglandular adenosis, and I'll come back to it, because in the textbooks it's down as benign, but it has all these malignant features. It's infiltrating through the stroma. There's no other characteristics there. It should be an invasive cancer, but it's not. The techniques that we learnt in research were a technique called immunocytochemistry, which is where you've got an antigen here, you produce an antibody to it, which sticks onto that, but then you put a coloured label onto this antibody that's sticking onto the protein that you're interested in. These are just basically proteins. And by seeing that colour, you can tell where the protein is. And you can do some wonderful things. I went to see Chris Elston shortly after learning about lymphoma immunocytochemistry and said, is there any research I can do in breast disease? And he said, oh, yes, probably. I'll talk to Roger. And they came up and said, well, we're working on this antibody that we produce to metastatic breast cancer cells. And this, I have to say, was the most exciting time in my life with respect to research, in that Bob Baldwin from the Cancer Research Campaign Laboratory said, well, yes, we're struggling to characterize this antibody. If you can help us, then please do. And I was able to get technical support from the lab at the time, and we started working with this antibody. These are gastric parietal cells, and this is the intracanalicular system of gastric parietal cells, which is where the acid is secreted that makes its way out into the stomach. Now, I had never seen that. This antibody just lit up this organelle in these cells, and it did this also strange things in lots of other organs, and there isn't time to go into that in detail. But every day I'd walk into the lab, look down the microscope and see something beautiful and new. And it is wonderful looking at things down, and you can play tricks with it. This is a way of changing immunocytochemistry so it glows. And these are, again, intracanalicular systems in gastric parietal cells. But it's beautiful to do that, and it's just a wonderful experience of seeing something that you don't understand and seeing it evolve in front of you. And that's why I love research and asking questions like why. We applied this to breast cancer, and this is a breast cancer with this protein. The brown stuff is the protein, the blue stuff is just the cells in the background. This is a breast cancer where there's no protein. There's a little bit in the normal bits of breast here, and this is a breast cancer where there's lots of that protein. It's all over the place here. And we produced evidence that expression of this protein related to outcome in breast cancer. Patients who had a lot of protein expressing had good news breast cancers, and those that had little had poor prognosis breast cancers. And we published in the BMJ, and I was awarded the Medkai McCallum Prize for that publication many years ago. So a great thanks to the Medkai, and this prize is still ongoing. We also had some fun. This is the world's first case of plumber's knee. There's a plumber in Nottingham who used to have end pipes over his knee and ended up with calcinosis cutis of the skin, and we published in the BMJ. My aim at the time was to try and get a publication into the Christmas edition of the BMJ, and we failed. This was published in June, despite us asking the editor to publish plumber's knee, the world's first case, in the Christmas edition. One of my other passions, as you may know, is wine tasting, and I'm indebted to Ian Dawson for allowing me to take 5 days holiday as afternoons attending a wine tasting course in Nottingham, which was the early rungs in the Master of Wine sort of ladder of progression in the professional hierarchy of wine tasting in the wine industry. Through that experience, I met people in the wine trade in Nottingham, and I joined a wine tasting club with that group, which essentially performed blind wine tastings. And I learned how to do competitive wine tasting. And for my sins in the early 90s, I came second in Wine Taster of the Year and snogged Julie Goulden at the final event. I was actually beaten by a gynecologist, which still rankles, I have to say. And the only reason I think I was beaten is he had a greater disposable income to practice with his passion. But it's interesting that two members of the medical profession came first and second in this national competition. When I was finishing my training, the Forest Report was published. This is the report on breast cancer screening, and it recommended that breast cancer screening was going to should occur in the UK. And Nottingham was one of the pilot sites for that, with Roger, Eric Roebuck, and Chris leading that. And Nottingham became one of the leading centres, and a training centre was set up here. And I was appointed as a consultant pathologist to work in that training centre and work in breast cancer. This is the Helen Garrett breast screening unit. It's porta cabins when it first was set up, and we'll see what it looks like now in a moment. It was sitting down here, but those early years working in breast cancer screening were fabulous in terms of developing technologies for patients and developing new experience and improving diagnosis. We were a training centre, so we were teaching the rest of the country how to do this, but we had to learn first. And we learned from masters in Sweden, in the States, elsewhere in Europe, and in the UK, and working with people like Rob Wilson, Andy Evans, Eric Roebuck, and Roger was just a fantastic experience of moving forward. I'm very grateful to the Medkai again, because they sent me off to the States and gave me a travelling fellowship to go and work with this man, who's David Page in Nashville, who's one of the gods of breast pathology, because I was reasonably seen as being reasonably parochial, particularly by this vocal individual, who's Tom Anderson, who is another god of breast pathology from Edinburgh, who felt that I'd spent far too much time in Nottingham and I needed to broaden my wings and fly elsewhere. So I worked with David for a short time as a visiting fellow. He calls me his first fellow in breast pathology, because I was the first person to knock on his door to learn from him. And the Medkai supported that. In 1978, I was appointed as a consultant here, and there's too little time to talk about it in detail, but I have a service teaching and research role. What I'd really like to do is to talk about my research for the last few minutes of my presentation. Because that's what drives me. I love to ask the question, why? Why does something look like it does? Why does it behave like it does? And then how does it do that? What's actually driving it? What's the underpinning process that causes something to do what it does? Nottingham is famous for breast research, and I take no credit for that at all. That's Roger and Chris who are the pioneers of that. They were appointed as consultants here in 1973 and set up a long-term sort of follow-up study of patients, and we have the tissue of those patients. I'm indebted to the patients who've donated their tissue for that research program, because I'm absolutely reliant on that material. I started medical student in medical school in 1973, as you remember. This is a breast pathology report that Chris found in the City Hospital files around that time. This is 1976, but this is a genuine report. The names are changed, and this is Nellie Bristol here, and Mr. Carver is the surgeon. But this is the actual general report. It just says, section shows spheroidal cell carcinoma of the breast. Now, that doesn't tell me anything as a pathologist, it doesn't tell a surgeon anything, but that's where pathology was in the 1970s with respect to breast. There was nothing else you would add to that. A patient nowadays couldn't be managed with that information. I'd be laughed away from the MDT if I presented just that statement. Pathology reports in breast pathology now are several pages long with lots of information that's essential for patient care. But why is that? Why has that happened over the years? Oop, I've got a frozen computer. Don't know whether my colleague's there. I don't know whether... So what we tried to do over the years, and certainly Roger and Chris were fundamental, but I came in to try and troubleshoot aspects of that in my sort of early years as a trainee in pathology, and then later on as a consultant pathologist. And the reason for that is, the reason that we were looking at breast disease is that from this very old slide, now this is the normal population life expectancy, and as somebody pointed out, life is a sexually transmitted university fatal disease, so this normal population curve does keep going down because we will all eventually die. But this is the breast cancer curve here, which separates continuously all the time from the yellow curve. So this white curve is going downwards, and it's going down steeper than the yellow curve. And this separation between the two is occurring at every interval, implying that women are dying from breast cancer 1 year, 2 years, 5 years, 10 years, 15 years after diagnosis. If you were ever cured, the curve would change. It would go down, then it would plateau, and it would mirror this other curve, which it doesn't do, which implies that the prognosis of patients with breast cancer is hugely variable. Hypotheses were that it was governed by groups of different variables, how long the cancer had been there, and the biology of the disease, what the cells are thinking about, what they're trying to do. Roger's early work in prognostic factors, and this is the first time multivariate analysis had been used in cancer in the UK. The time lymph node stage was believed to be the most important prognostic factor in breast cancer, whether the disease had spread to the lymph nodes. But we demonstrated that tumour size, lymph node stage and histological grade were equally important, and that you needed all of that information to be able to govern providing a patient with information about her prognosis. Now in the Royal College of Pathologists we have a whole set of different variables we look at with respect to governing prognosis in breast cancer. But if we start to tease out those prognostic factors, it's fascinating because we begin to see and make observations that actually put some meat onto why we need more than one piece of information to decide how a patient will do in the long term. If we look at histological grade, we separate into 3 grades, well differentiated or grade 1, moderately differentiated and poorly differentiated. If we look at the grade 3 curve, it has that interesting phenomena of plateauing. So patients with grade 3 breast cancers die up to about this point ahead of other patients, and that's about 7-8 years. And after that, it's actually quite rare to die of a grade 3 breast cancer. Grade 2, this happens about 12-13 years, and grade 1 probably happens about 25 to 30 years down the line. And it's interesting that the grade 2 curve is plateauing about the same proportion of women who've died, about 40 to 50%, as the grade 3 curve. So what it's telling you is that if you have a grade 3 cancer, you will die. If you're going to die more quickly. And if you've got a grade 2 cancer, that process will be slower. But the same proportion of women will die whether they have grade 2 or grade 3 cancer. So it doesn't tell you whether you're going to die or not, because the same proportion of women will die regardless of what grade. And that was shown by this curve here. I'll pass on from that one. Tumour size, another of the prognostic variables, is interesting because we don't see that plateau effect of these curves. And what tumour size is telling you is not when or not the speed of events, but it's telling you your probability of death occurring. And this is how breast screening works. If your cancer is larger, then you've got a greater chance of dying from the disease. If it's smaller, you've got a lower chance of dying from the disease. But it doesn't tell you when that is going to happen or how that will happen and the behaviour of that process. So it's telling you different features. And interestingly, lymph node stage probably tells you more like, well, it tells you more like the size of the cancer. It's not linked to the speed of events largely. What we were able to do and what Roger had started doing was to develop an index by putting these together, and this was able to stratify patients across the whole spectrum of outcome in breast cancer, where we could identify patients whose prognosis is exceptionally good, and this is as good as patients without or people without breast cancer versus those that have an extremely poor prognosis at the bottom. But at the time, Roger was going around the world saying, well, this is how we should manage our patients with breast cancer. And other people were saying, well, we can't grade breast cancer. It's too difficult. Those people in Nottingham, the pathologists you work with, clearly have some form of witchcraft they're using to do this, but we can't get it to be reproducible. Roger just said, you pathologists, you're cantankerous and difficult, and you don't know what you're doing. At least the other ones don't, but you might do, but I don't know how you do it. But Chris and me then sort of, because I was just a youngster in the lab, we were sitting there thinking, well, how can we solve this? There's no doubt that grading works. There are a huge number of publications in the medical literature that showed that breast cancer grading had worked. And so what we set out to do is just to put some very simple objective criteria to it. I won't bore you with the details of that, but essentially we looked down the microscope and compared normal cells with the breast cancer cells, and we were able to do things like counting mitoses in detail. Just very simple things that you can do down a simple light microscope. And we were able to then develop what we believe to be a more robust and reproducible grading system. And that met with satisfaction. There were groups elsewhere in the world, in Australia, America, and Europe, who produced reproducible results using this methodology. And it's now become the WHO, European, UK standard for classifying and grading breast cancer. So we've managed to go just from being sort of demonized by Roger and saying you pathologists don't know what you're doing, to having a standard that works. So Chris has really, he's got a better hairstyle now, he's got a better microscope now, and he's a smiling man. And sadly he's not here today, as I say, he's away on holiday, but great credit to him. Roger had a party to celebrate that grading was now reproducible in the world. Didn't invite any pathologists, they're all surgeons in that photograph. Chris, interestingly, got some fan mail, and I'll just put that a little bit higher. Somebody believed that Dr. Christ Elston's intervention was essentially something from above, and it should be acknowledged in that way, and some sort of divine process. Lots of other things we did too. I'll just go through this fairly quickly, but this is looking down the microscope. Just imagine these are wallpapers. These are different types of breast cancer, but they all look different. And they all behave differently. The first one was a mucinous carcinoma. This is a classical lobular cancer. This is a tubular cancer, and that's a garden variety ductal cancer. They have names, but they also have different behavioral characteristics. And this is Tom Anderson's work from many years ago, showing that patients who lived long term after breast cancer diagnoses tended to have these funny types of breast cancer, where those who died quickly had the garden variety of breast cancer. We also showed similar data with excellent types here. Tubular cancer is one of those. And it's interesting that these observations, do they have any clinical relevance? Well, yes, I remember a corridor conversation with Roger saying, well, he said, I don't think anybody we've diagnosed with tubular cancer ever died of their cancer in Nottingham. I said, well, that's important. Can we study it in more detail? And he said, no, because I don't think we've had enough. But a few years ago, after Roger had retired, Emad, my colleague, and I sort of followed up that corridor conversation, and Emad looked at all the tubular cancers we'd had. We had about 100 by that time, nearly 3,000 breast cancer cases, and we were able to compare those with other similar types of breast cancer. And what we were able to show is that the patients with tubular cancer did not die of their disease. These are so slowly growing that you will die of something else before it catches up with you. But should you still follow up a patient with this cancer? Yes, because they've got a risk of getting another cancer. And the few deaths we've had with tubular cancer patients, they've all had second breast cancers that have been higher grade and died from that second breast cancer rather than the tubular cancer. Back to immunocytochemistry, because we spent many, many years working with immunocytochemistry. It's a fantastic tool. What it does is allow you to look at the tissue and see where the protein is being expressed. And this brown pigment is HER2, the protein that's the target of the drug Herceptin. And we worked with Bill Gullick right in the early years when this was first discovered, not the drug but the protein, and working and demonstrating it was a prognostic factor. If you had expression of HER2 protein, then your breast cancer was worse, long-term survival less than 20%, and here if it's not expressed, then a much better survival. Now there's a drug to produce to that. It's interesting that Bill Gullick tried to persuade the MRC to develop a drug to that target, failed, whereas in the States Dennis Lehman was able to develop, progress the drug, and Herceptin was developed in the States rather than the UK. But Bill Gullick, I think, had the first idea. We looked at numerous proteins over about a period of 10 to 15 years, and Roger was convinced that by identifying single proteins he could get rid of those cantankerous pathologists, because looking at proteins was objective, they were present or they were absent, and you didn't have some subjectivity of a pathologist looking down a microscope. But I have to say it all failed, and we coined this term a YAPI, or yet another prognostic indicator in breast cancer. And we looked at virtually every protein that's known to be of relevance. We demonstrated it was prognostically relevant in breast cancer, but it wasn't relevant in the clinic setting because grading was actually better than looking for any of these proteins. Surprising, but interesting. Molecular biology then emerged as a tool, and I felt that breast cancer should be the same as things like colorectal cancer, where a sequential pathway had been identified going from normal cells through adenomas, through carcinoma in situ, through invasive cancer to metastasis, with different genes being involved in each process. In breast cancer, we couldn't identify any genes associated with that progressive pathway. If you looked at breast cancer as a whole and just put a group of breast cancers into a genetic analysis, virtually every chromosome came out as being involved. But it's only when we started to look at individual types of breast cancer, and this is that tubular cancer I mentioned before, that you actually saw very specific genetic alterations, inferring that breast cancer is not one disease. It's actually a group of different, molecularly very, very different diseases. And when you begin to appreciate that, then you can begin to tease out the genetics of breast cancer. And that molecular biology taught me another thing too. We have dogma and we have beliefs, and sometimes new data challenges those. So in the breast cancer literature, we have lobular breast cancers and we have ductal breast cancers, and we have low-grade and high-grade ductal cancers. And we have textbooks. Chris and me have written a textbook in breast cancer where we have the ductal cancer chapter and we have the lobular cancer chapter. But when you look at the molecular biology, the low-grade ductals have the same alterations as the lobular cancers, and the high-grade ductals have a completely different set of molecular alterations, implying that we've got the whole thing wrong. That was further supported by the gene chip analysis. This is a beautiful set of data. I remember this being first presented by a guy called Chuck Peru from the States. This is felt to be groundbreaking and sort of life-changing research. He presented it to a meeting of a breast cancer meeting in Brussels, the big European breast cancer meeting, thousands of people there, and he said this was the end of pathology. Full stop, end of pathology. He's a molecular biologist, doesn't care about pathologists, end of pathology. So we had a conversation, a few pathologists met outside, and there were some very experienced pathologists there, and you could see the cup half empties versus the cup half fulls. The cup half empties believed this was the end of pathology, and they were going to the bar now, and that was it, and they were going to get a new job. And there's the other ones, I'm a cup half full. I just said, well, Chuck Peru doesn't know what to put onto his chips, and I do. And I know if you put rubbish onto a chip, you're going to get rubbish out. So at least I'll have a job putting things onto chips. But of course, this has not stood the test of time, whereas grading is still in our armamentarium. There's been a few spin-outs from this, and I'm going to show you one or two bits of data. And there's a beautiful example here of putting rubbish onto a chip. In the first publications, there was one of the molecular classes, they're all in different colours here, but this green class is called normal breast-like. And when they went back many years ago and looked at the tissue samples that they put onto the chips at that time, the tissue samples didn't contain cancer, they just contained normal breast tissue. So they were putting rubbish onto the chips and getting rubbish out, and it's in this major publication in Nature. Does it work as well as grading? No, this is stratification using gene chip analysis, and you can separate good prognosis patients from bad prognosis patients, but it's not as good as the Nottingham index, which is free, available from the data that you get from your routine pathology reports. A gene chip analysis will cost you several £1,000s. But there are things you can do with this, and here's our tubular cancer again. It's one of my favourite cancers. It's beautiful looking down the microscope. You have to believe me because I'm a pathologist. It does look beautiful to me. But by using this type of methodology, you can start to do interesting things. You can start to dissect out the cancer cells and look at them in their molecular sense. And you can dissect out their cohabiting benign and other diseases in the nearby breast tissue. And if you begin to do that, you can map out what might be a precursor lesion. In the textbook of breast pathology I started learning in, as a Pardy's textbook, it said there are no known precursors to human breast cancer. And that's another one why there must be. In the colon there are, in the lung there is, in the cervix there is. Why isn't there in the breast? And so that's one of the things that's driven me over the years. And so using this molecular approach, and this is a gene chip analysis, but done where the genes are arrayed so they go across the chromosomes. So we're going from chromosome 1 here right across all the chromosomes. And this is essentially known as a genome plot. And if you go up, you've lost. If you go down, you've gained in terms of expression of a part of a chromosome. And you can see by mapping out this is a benign alteration next to a tubular cancer, then there's carcinoma in situ, then there's a tubular cancer. And it's interesting that the benign lesion has some of these alterations that are present in the in situ disease and in the invasive cancer. The invasive cancer has more alterations, but you can see this progression going through. And that's good evidence that these lesions are the precursors of some forms of breast cancer. Now, this is one of my most favourite and beautiful cases. This is two types of in situ breast cancer coexisting in the same duct. The paler cells here are lobular cancer, and the darker cells are ductal carcinoma cells. You have to believe that, but that's what I'm saying. But if you don't believe me too much, there's a protein that's associated with lobular cancer, and that's lost in lobular cancer, and that's present in ductal cancer, and that's the E-cadherin protein. Using immunocytochemistry, the lobular cells are negative for that protein, the ductal cells are positive. So is that a collision of two different lesions, two different entities, two different cancers coexisting? In the textbooks, it would lead you to believe that they are completely different. But if you do this genome plotting, the adjacent benign disease, the DCIS, and the lobular cancer all have the same alterations, and they have a cyclin D1 amplification that goes through, implying that these are the same entity that have grown in the same way. A further single gene hit on one of them has changed its morphology and makes it a different disease process that looks different down the microscope and indeed behaves differently. So by applying this type of technology, you can actually answer questions that have been dogging you for many, many years, and also questions and uncertainties about how you classify various things. The last couple of minutes, I'm just going to go through where things are going in the future. One of the passions, if you're a scientist, is to get a publication in Nature, and I managed it. And this was a collaboration with a group in Cambridge, and I'm really indebted to them because they essentially did all the work when we provided some of the cases. But this is, again, following up from that molecular analysis of breast cancer using a very, very complex approach. Now, this is never going to translate into clinical practice in terms of a test diagnostically, but may translate into new drugs by understanding the biology of the disease. Two different breast cancers here. If you just look at this survival curve, green, this one purple or pinky purple, they're both estrogen receptor positive breast cancers. These patients do very badly, less than 40% alive in the long term. These do extremely well, best surviving group. But they're both principally the same molecular classes. But if you start to look at the molecular biology, they have very, very different entities. The good prognostic one has low genetic instability. The poor prognostic one has specific molecular alterations, which you could begin to tease out using bioinformatics. I don't do bioinformatics, it's far too complicated. But this is mapping out that gene expression profiling, looking at how the genes signal to each other and how they can influence a cancer cell. And if you can understand that process of gene signaling, you can actually work out ways to interrupt it and hopefully stop the cancer growing. Final thing I'm going to talk about is deep sequencing, which is just on us. David Cameron, one of the good things he's done was to fund the sequencing project, the 100,000 Genomes project, which is looking at 50,000 patients with cancers. Gene sequencing now is a fantastic technique. It's available, it's relatively cheap now, and looks at the mutations and other genetic alterations in cells. I won't go to the details because there isn't time. It's far too complex and I don't really understand it that well either, but you can map out the genetic alterations, and these are just 3 breast cancers here, just showing completely different patterns of disease. You have to believe me that these are different patterns of mutations, translocations and other genetic alterations. But you can do this now very, very, very quickly. And what it's doing is improving our understanding of the biology of breast cancers and other cancers, and demonstrating that cancers are heterogeneous. They're not a single cell that's developed. They're generated from one cell, but then you develop, through probably a Darwinian type of evolution, a family of cells. And this was shown by a guy called Charlie Swanton a few years ago, looking at renal cancer, where a patient donated their body after for post-mortem, and then he was able to map out the genetic mutations of this metastatic renal cancer. And what he was able to do was to look at how different clones of the tumour progressed in this sort of phylogenetic type tree, just like Darwinian evolution, as shown there. And this is that microglandular adenosis case that I showed you earlier on, and only last year we were able to crack this nut, and it's taken 35 years to do this. But this is microdissecting out microglandular adenosis, and we produced a few papers over the years showing that these benign entities are associated in some circumstances with progression to a particularly aggressive form of breast cancer. And with my good friend in the States who got all the toys that we haven't got, we were able to deep sequence microglandular adenosis cases demonstrating that they do have fundamental genetic alterations, and they're actually the early precursors of particularly aggressive triple negative types of breast cancer. So it's interesting that you still have nagging things in your head, and it may take you 30, 35 years to be able to resolve them, but your doubts were correct in the first instance, and that was proven last year. So what does the heterogeneity mean to us? Well, as a pathologist, when we look at a cancer, we look at a snapshot of what is there at that time in that patient. But if we look at the evolution of a cancer over time, it probably does have this Darwinian type of evolutionary process. So if you look at this tumour here, it starts off as grey cells, but you get this purple clone emerging, then a blue clone emerging, a darker blue clone emerging. This one dies out, that one dies out, this one continues to grow, other ones grow. And if you take this snapshot of a biopsy at this time, you don't know what's going to happen then. You don't really know what's happened in the past. All you know is what's going on at the time. This could be where, and this is where our research is going at the moment. We're trying to do this in collaboration with a group in Saudi Arabia who have lots of money and all the technology. But it's important because if you've got a heterogeneous tumour, you may have a group of cells in your cancer. The blue ones won't respond to your treatment particularly well. The green ones will respond okay, and these ones will respond very, very well. And if you give a weak treatment, then you may lose some of them, but not all of them, but the blue ones will grow out. If you give a very strong treatment that kills all of these ones, you'll just end up with those cells. And so the hypothesis now is that if you can actually look at the heterogeneity, look at the patterns of cells in your tumour, you can predict what the resistance or the resistance outgrowth of tumours will be in time to the drugs that you're going to give. You can identify what they're likely to be, and you can give second-line drugs either early on or anticipate what's going to happen and try and identify that earlier on. So I think heterogeneity is where the future is going to be in terms of the type of research I do. I think I've probably bored you stupid with what I love, which is research in the biology and molecular pathology of breast cancer. But what it's done is, over the years, it's taught me why breast cancer looks so different. These are all breast cancers around the outside here, and that's a normal breast lobule. But it's really fed me with solace that there are explanations for the differences in what I see down the microscope and also how cancers behave. I was fortunate to be appointed as president of the Pathological Society in Great Britain, and we were able to host the largest cellular pathology meeting in the UK, nearly 4,000 delegates in London a couple of years ago. And this is the team there. This is Imad, my colleague, Abik, who's just been appointed as a consultant pathologist. There's the old Git in the middle, and these are all our students. There's Andy, who's the scientist, and Mohammed, the scientist there. So it was wonderful to be able to take our team to that meeting and just talk about all the things we love doing. Chris, as I mentioned, can't be here, but he now spends most of his life playing golf and drinking wine at Hearts with me sometimes. Roger, sadly, who I have to say, Chris and Roger were my real mentors over the years, though there are many, many others. Roger, I dearly miss. He died, as you may well know, a short time ago, but he was a true inspirational character, a visionary surgeon, and somebody who appreciated teamwork and supported individuals. I remember when I first presented that work on NCRC11, the breast cancer antibody that won the McCallum Prize, Roger wrote to me afterwards and said, just a written note, say, fantastic, well done, and a pat on the back. I was just a lecturer at the time, and Roger was a professor, and it was just a great thing to do. Complete contrast to Curran, who I mentioned right at the beginning, who said, I will kill you if you walk into my lab. What a difference, absolutely unbelievable. Roger, now his legacy is in the lecture theatre in the Breast Institute. There's also a legacy of his ties, and I meant to go and photograph, but Norma, his wife, Roger loved colourful ties, and just through that window on this wall here, there's a collage of Roger's ties, and I commend you to go and have a look at that. It's wonderful. His ties were really, truly great works of art. I also commend to you the programme. I won't go through the details of that. Many of the talks are relevant to me in my life, and there may be some explanations that I've given to you earlier on. And I'll leave you with just one final pathology message. This is a prostate biopsy talking to a breast biopsy. And so pathology is fun and beautiful at times and can make you smile. Thank you very much for attending tonight.
Well done.
Wow, I mean, that was amazing. I don't think you can hear me for that. Sorry. Wow, that was absolutely amazing. I'd just like to thank Professor Ellis for the absolutely fascinating talk he's just given, giving us insights into his growing up in medicine in Nottingham. We've talked about Lancashire, Yorkshire, lacrosse, fish, budgies, beautiful cells glowing. It gives a lovely image. As someone who also grew up in medicine in Nottingham, it's really good to hear about how different paths can be carved in medicine in Nottingham. I feel quite lonely in your presence. It's a tremendous credit to the university that such an internationally acclaimed professor as Ellis is grown from here. I would personally like to thank you for all your words of wisdom that you gave me last year, and I look forward to your exciting programme of events, which I feel quite sure that you'll all support many members. So ladies and gentlemen, I'd like to wish Ian every success in your presidential year, and formally thank you for such an intriguing and illuminating talk, and ask you to express your appreciation in the usual way.
Thanks for your help.